Hello, I am John Martin. This video is about the incorporation of bacterial genetic sequences in a stealth-adapted virus cultured from a patient with the chronic fatigue syndrome. The origin of the virus was from the cytomegalovirus of African green monkey. It's called African green monkey simian cytomegalovirus. This was an unexpected finding. It has relevance to several common illnesses that are currently being attributed to a bacterial cause rather than probably being viral in origin. These include chronic Lyme disease, Morgellons disease, Pandas, and others. The other aspect is it's biologically very interesting and has huge implications if the bacteria are able to transmit stealth-adapted viruses. Now, the next three slides I've shown in previous videos, which I hope, if you haven't seen them, you will review the earlier videos. The first photograph is that of the positive culture from the chronic fatigue syndrome patient. These are stained cells, and they show the marked cell damage that can occur. But it was possible then to use the polymerase chain reaction to amplify portions of the viral genome. This showed the results from the polymerase chain reaction performed on the positive viral culture. There were products that were amplified, shown in this band, and I could label these with radioactivity and then look for virus in the supernatant or the fluid from the viral culture. This shows that we could identify and isolate DNA from the positive viral culture and show it in, this is an agarose gel, and the distance the DNA migrated down showed that the DNA had a size of approximately 20,000 nucleotides, but we also know there are multiple segments or multiple sequences in this band. With that DNA, I could clone it into plasmids. These are ways in which small segments of the DNA can be incorporated. They can be used to infect bacteria, amplify the DNA. I used two of what are called restriction enzymes, EcoR1, SAC1, to give overlapping fragments, and we had well over 200 clones that were partially or fully sequenced. This goes back to some of the earlier data that I presented. 200 of the clones match closely to genetic sequences within this African green monkey simian cytomegalovirus. It was interesting. There was an uneven distribution along the entire genome, with multiple clones matching to some regions, but no matching at all to other regions of the cytomegaloviral genome. The size of the cytomegaloviral genome, when it's intact, is 226,000 nucleotides. And we could show overall that the clones covered or were matching to approximately 100,000 nucleotides of the entire sequence. The other parts of the virus were not detectable. Therefore, going back to the fact that the DNA was migrating as if it were 20,000 nucleotides, we have at least 5 fragments if we take into account there were at least 100,000 nucleotides. What we also showed with this part of the study was that even for clones that matched to the same region of simian cytomegalovirus, there were a lot of internal differences. There was therefore genetic instability of the viral-related sequences. I published these data in 1996 in this article, Genetic Instability and Fragmentation of a Stealth Viral Genome. What I'd like to do now is focus on those clones that had bacterial sequences. In the last video, I described these, the 14 clones that had cellular-derived genetic sequences, but we had 34 clones, quite a significant number, that had bacteria-derived genetic sequences. Now, I was confident that there was no bacterial contamination of the cultures. I had maintained the culture from this patient in antibiotic-free fluids over long periods at different times. There was no overgrowth or outgrowth of any bacteria. I've also had several occasions, like I showed earlier, where we could look at stained cells from the culture and there was no indication of any bacteria being present. I even did multiple electron microscopic studies on the cultures, and again, there was no indication of any intact bacteria. Also, when we have analyzed the sequence, they've diverged from the standard bacteria that one might expect in a contaminated culture. Finally, in terms of the SAC1 restriction enzyme, the DNA was obtained not only from filtered and ultracentrifuge culture fluid, but had also been passed through agarose, and I selectively took the 20,000 kilobase band, which is far smaller than any bacterial DNA if it were contaminating bacterial DNA. I therefore felt quite comfortable to introduce the term Bacteria for viruses with incorporated bacterial sequences. These early data were published in an article in 1999, Bacteria-Related Sequences, in a simian cytomegalovirus-derived stealth viral culture. Now, all the data we had, we submitted to GenBank. GenBank is still growing. At the early time, 1999, there wasn't that very much bacterial sequences. So I can continually upgrade the matching as more bacterial sequences are being submitted to GenBank. This is an article in 2019, and it updates some of the matching data for the bacterial genetic sequences. Also note here, I've used this term renegade for both the cellular and the bacteria-derived sequences. They're like sequences hitching a ride with the virus to become part of the virus and then be carried from the virus, from monkeys to humans, or from the bacteria into human. The next topic is the types of bacterial sequences. Of the 34, most of them were related to a broad group of gram-negative bacteria called a-Proteobacteria, early Proteobacteria. But the best matching occurred to members of the species Onchrobactrum, which I'll describe in a moment. We also had sequences from Mycoplasma, Microbacterium, and still we have sequences where we have yet to identify their origins. This shows examples of very good matching to one particular species of Onchrobactrum, the Coromnosans. Clone 3b23 has 8,916 nucleotides long, and 8,911 of them are identical to those in this particular Onchrobactrum, 99%. Another example, clone C16144, 4,142, and with one space in it, 4,127 of the 143 match to this particular Onchrobactrum. Yet we've had other clones, 3b513, quite a long clone, and there's no significant matching to this Onchrobactrum, even though it's still, this particular one has features of an a-Proteobacterium. Now, where the Onchrobactrum fit in is that they're related to Brucella, and in fact the genus has a common name, Brucella Onchrobactrum. they have identified 12 species of Brucella, mainly infecting different types of farm animals, though many of them can cross and cause a undulant fever in humans. There's more species, 18 of Onchrobactrum, that have been described, but in reality, there are many, many more species that have yet to be described. The big difference, Brucella are associated with animal infections, Onchrobactrum are associated with infection of plants. This is why the family name, both terms, Brucellaceae, or the Rhizobiaceae, as the Rhizobialis in the order refers to the fact that it's more of a plant-type bacterium. And that's all within this class of a-Proteobacteria. What I should address now is where can the virus acquire these bacterial sequences? The first obvious one, if there were co-infection of the cells with viruses and bacteria, then there could be a similar mechanism whereby the virus can incorporate cellular sequences. The same within the same cell, there could be bacterial sequences. The most likely explanation is that these renegade sequences cross-link fragments of the viral genome so that they can then be incorporated into the virus. The more interesting suggestion is stealth viral infection of bacteria. What I know about the virus, this particular virus, I could infect many types of cells from many different species with the virus. It was pretty impressive that whether it was rabbits or duck or mice or humans, monkeys, all the cells were susceptible, and even insect cells could be infected with the virus. A very wide range of host susceptibility, which is not a characteristic of most viruses that are restricted, but they're restricted because they have restraining genetic mechanisms that may be lost in these stealth-adapted viruses. So it's not unreasonable that they could even extend their ability to be infectious within bacteria. Moreover, I had the occasion to test bacteria from the feces or the stool of the patient from whom we had the positive culture from the blood. There were very interesting bacteria in the stools. They were abnormal, atypical, and I could even take extracts from these atypical bacteria and show that they were positive in cultures, though I never went ahead and fully sequenced or did any sequence data on the bacteria isolated from the viruses isolated from the bacteria. Now you can ask, well, where would the bacteria come from? These Onchrobactrum can be part of what are described as food-derived soil bacteria that come in with the food that either humans eat, or in the case of the African green monkeys, would be eating food that would have some of these soil bacteria. So that could be the source of the bacteria. Now it has huge implications if stealth-adapted viruses are infecting bacteria, because they could then transmit the infections from one person to another, and it's much more difficult to eradicate bacteria from the environment than viruses. The next big question, why would the virus acquire these bacterial sequences? One suggestion, they could serve as additional proteins, like a capsid protein, to help protect the DNA as it passed from one cell to another or from one person to another. There are certainly metabolically interesting genes in the bacteria that we've identified. They could be providing additional energy pathways to support the viral replication, or if there are infecting bacteria, energy pathways to support the bacterial replication. Indeed, what I feel is a reasonable suggestion that still has to be pursued is what we've described as alternative cellular energy pigments, materials produced by these viruses that have unusual structural characteristics. Some of those could well be these bacterial-derived products, and they can act as a source of energy, not necessarily for the virus or for the bacteria, but for the cells, and can act as a non-immunological defense mechanism to help protect the cell. Now, the next big topic on this one is to address the issue of several illnesses that are attributed to bacterial infections, yet they have many features in common with a stealth-adapted viral infection. First of all, just to go back, the chronic fatigue syndrome and the Gulf War syndrome were previously attributed to Mycoplasma and Brucella. One example of that was an article back here in 2005, and other people have similar data where they could do molecular assays, not particularly precise, but molecular assays, and they could find evidence of Brucella, Mycoplasma species co-infecting patients with the chronic fatigue syndrome. It even went further with a gentleman in Canada who I met, Donald Scott. He had written a book, The Brucellosis Triangle, and in the book he linked fibromyalgia, chronic fatigue syndrome, Gulf War syndrome to a laboratory-engineered form of brucellosis. It's known that brucellosis has been modified as a biological weapon, and he thought that was a spillover into the cause of chronic fatigue syndrome. On the East Coast, many patients who have a chronic fatigue-like syndrome are being told they have chronic Lyme disease based on some antibody and some molecular analysis finding genetic information of Borrelia burgdorferi in the patient. Dr. Borrascano, who's one of the leaders in this grouping of chronic or treatment of chronic Lyme disease patients, would send me really several hundred cultures that were invariably positive for stealth-adapted virus. So I, for other reasons also, believe that when the stealth-adapted virus is infecting a cell that's co-infected with Borrelia, which grows inside cells, there could be that genetic exchange. There's another illness, Morgellons, and it is, in my mind, the overproduction of these alternative cellular energy pigments. They're electrostatic, they jump about. The patient quite reasonably attributes it or sees these materials as parasites. But the arrogance of the psychiatrists and dermatologists is to say these patients are delusional. They're mistaking just regular environmental material. And they give an unfortunate diagnosis, delusional parasitosis. But again, the mainstream people pursuing Morgellons see it now as a Borrelia infection because there are some Borrelia sequences in some of the Morgellons, some of the patients diagnosed with Morgellons disease. Again, from my own studies, all the Morgellons patients who have tested are stealth-adapted viral infection. Another illness that's worthwhile including, PANDAS. This is referring to a pediatric or childhood autoimmune neuropsychiatric disorder associated with streptococcal infections. I've had the occasion to test several children diagnosed as PANDAS. What was interesting to me is when I did the streptococcal testing, there was often discordant results using different assays for streptococcus. And again, it needs to be pursued to see if this is not a streptococcal infection, but a stealth-adapted virus that's incorporated streptococcal sequences. And we can even go to studies that have looked at Porphyromonas gingivalis as a type of bacterium that we have in the mouth is being linked to Alzheimer's disease. But there's not so much the whole bacterium, it just could be incorporation of some of these bacterial sequences. Now, with that in mind, I just want to emphasize it's so important to establish a viral culture and sequencing facility to study stealth-adapted viruses. Thank you for your time.